Hello student, this is the continuation part of the last video. In this video we will discuss about the rest of the steps of the for the formation of recombinant DNA. So let's start with after formation or after transformation of the cell of or after transformation of recombinant DNA into the host cell, it needs to be screening. Now how we get to know that how we get to know that all the clones we get are transformed cells. So to differentiate from transformed cells, those who get a foreign DNA, and non-transformed cells in which foreign DNA is not present, some techniques are used which is known as which is used. These techniques mainly include the antibiotic resistance or some screening test. Antibiotic resistance is provided due to the with the help of vector. It depends which vector we are using. Some vector, like I told you, PBR322 contains two sites which are ampicillin resistance and tetracycline resistance. So those DNA, or those transform cell which takes the foreign DNA and replicate and make clone, there must be penicillin, ampicillin and tetracycline resistance. So if we grow them in a media containing ampicillin or tetracycline, only transform cells will grow and non-transform cells will not grow. In this figure you can see there are some blue colonies and there are some white colonies. This is the method in which beta galactosidase enzyme is used. So those who are transformed cells who take up the foreign DNA, they will form some blue colonies, and those who have not take up the foreign DNA, they will form the white colonies. So, and this is the method to identify or to screening the transformed cells from non-transformed cells. After identification of transformed cells and eliminating these non-transformed cells, we proceed further. So these are the main steps, and screening is necessary so that we can get false result uh, at further stages. So screening can be done either by using antibiotics or by using beta galactosidase enzyme. Now, obtaining gene product. So for obtaining gene product, the ultimate aim for this is, or for ultimate the ultimate aim for biotechnology is to produce protein, desirable protein like insulin. Okay, so the culture is used for extracting and purifying the desired protein, and these reactions are mainly carried out in the bioreactors. Bioreactors are large big tanks and with these reactions that takes place. Now as you can see, this is a bioreactor and it is made up of steel, and here is a media which need to be grown because we need our desired product in large quantity. So when we need our product before its marketing, because we need it in large quantity, we need it to be multiplied large scale. So bioreactors are used. There are two types of bioreactors are used. This one is simple stirred tank bioreactor. In this you can see this is sterile air. Even this air which is passed into this bioreactor is sterile, means it does not contain any microbes, any bacteria, so that our broth, culture broth, does not get harmed or does not get infected. This is culture broth. These are flat bladed impeller. What is their function? They rotate so that the mixture in this it's also rotates. So basically they are mixing the culture broth. Okay, and this is a foam baker. Foam baker means when this culture broth will mix, it will make some foam. So the this is a foam baker which is used, which separates the foam from the rest of the media, and this is the motor for the rotation of the this stirrer or impeller. And this is acid base control, because there are different drugs of protein which functions at different uh, acid and pH maintenance is very necessary. If it is basic, then pH must be in range of 8 to 14, and if it is acidic, its range must be in 1 to 6. So pH indicator is very necessary. And steam is here you can see steam is passed for sterilization. Sterilization is a process of removal of microorganisms or the bacteria so that our broth won't harmed. And so this is the bioreactor, which is made of steel, and this is motor which is impelling. So the main function is to get our product in large scale, and for getting it bioreactors are used, and all the required things are in this bioreactor, and it is being mixed, and and the sterility conditions are maintained. Okay, and next is sparged stirred bioreactor. In this this is a broth, and gas is produced. The bubbles dramatically increase the oxygen transfer area. Here this is the impeller. Okay, this is the impeller which rotate the media, and from here the oxygen is or the gas is produced, or the passed from there. So this oxygen is produced and air, so that it is used, so that the mixture get mixed easily. And here you can see this one is different from that one, because in this gas is transferred. Okay, and here there is a increased surface area, so that large amount of oxygen gets transferred. So these are the two bioreactors which are used to transfer to make our desired product, which was made by using our DNA technology at large scale. And after getting this, there is a last procedure in this chapter or this video, is this downstreaming process. In this downstreaming process, after the formation of the product which we are making, after the formation of that product, it is subjected to raised at a market. Okay, so for marketing, all the steps required for marketing comes under downstreaming process, and it includes the purification and separation, and these are kept in preservatives so that our product won't get uh, harmed by the fungi or any other bacteria or microorganisms. It gets saved by that. And sometimes clinical trials also run on drugs. That means if the product made is drug, then then it must be clinical trial or subjected to test on some animals, so that it won't harm people or the patients on which it is administered to. So clinical trial are must in the case of drugs. And here I'm ending this video. With end this video, the course is also ended. I hope you have learned a lot from this video about biotechnology process and steps. Thank you for watching my video. Thank you.